Hello, this is Dr. Pankaj Kumar, your mentor for today's session, and in this session we will be talking about the growth inhibitors, that is ethylene and abscisic acid, right? So first of all, let's talk about the ethylene. We know very well ethylene is a gaseous hormone, okay, and it stimulates the transverse or isodiametric growth, right? And it is very effective, that is in between 0.01 to 10 ppm, okay. And the bioassay for the ethylene is what we call triple response test. That is the name of the bioassay. Through that the ethylene is actually being tested, right? Let's talk about its biosynthesis. You know, ethylene is synthesized through methionine, or methionine is going to act as a precursor for ethylene, okay. And for its synthesis, what happens? Oxygen is essential, and you know, every cell is actually capable of ethylene production, but the maximum production occurs in the ripening fruit, okay, and that ripening fruit is actually called as a climacteric fruits. At the same time, it is also produced during the very process of senescence, okay. One more thing, what happens during the ripening fruits? There is a sudden increase in the rate of respiration, okay, and that is actually called as a climacteric rise. That is a very typical features that is always associated with the ripening of fruits, okay. Now let's talk about the ethylene functions. The important function of ethylene is the ripening of fruit. That is the most important function, but besides that, it also inhibits stem elongation, right, but promotes transverse growth, okay, and it also helps in the production of parthenocarpic fruit, okay. It promotes leaf abscission. One thing I would like to make it very clear, that regarding all other abscissions, it is the abscisic acid, but for leaf abscission, it is the ethylene that is going to promote the leaf abscission, okay. It also promotes senescence. So what is senescence? That is nothing but a programmed death. So that is also going to be done by the ethylene only, okay. Now apoptosis, it is a term that is used for a programmed death, because every cell has some sort of death of a cell is actually being programmed, okay. So apoptosis, what we call it is a programmed death of a cell is actually called as a apoptosis, okay. One more thing, what happens, that the application of ethylene on pineapple that induce flowering, okay, that is one of the another very important features of or commercial significance of ethylene, right. It also produces feminizing effect along with the auxin and cytokinin, okay. Now let's talk about the abscisic acid. You know, it also acts as one of the inhibitors. So it is going to act under the adverse environmental condition, and that is why it is also called as a stress hormone, okay. And what actually it does, it results into the formation of the abscisic layer, okay. So what happens, not only the formation of abscisic layer, it is also associated with the dormancy of many of the plants. Now let's talk about little bit about the history. The credit for letting us know about the abscisic acid goes to Frederick Adicott, okay. He first identified and characterized the abscisic acid. So they isolated two abscisic acid, one what we call abscissin 1 and abscissin 2, okay, but abscissin 2 is presently called the abscisic acid. You see, abscisic acid is absolutely identical with the substances that cause bud dormancy, okay. So initially it was called as a dormin, okay. So dormin is nothing but it is one of the other form of abscisic acid, right. Now let's talk about the biosynthesis. So it is produced in leaves from its precursor, what we call violozanthin, right, and is translocated by phloem to the stem. Now we know very well xanthozanthine is the intermediate of AbA biosynthesis, okay. So the structure of xanthozanthin or violozanthin indicate that they are terpene derivatives, right. So abscisic acid is found in all plants except the thallophytes. That means they are not going to occur in case of algae, fungi and liverworts. Okay. So in case of liverworts and algae, it is actually replaced by another inhibitors, and the name of the inhibitors what we call linolenic acid. Okay. Now let's talk about the ABA functions. One of the most important function of ABA is the formation of abscisic layer. So have a look at this particular diagram. You can see there, so this abscisic layer is formed, and before it gets parted away, okay, it ensures that the part of the plant should not remain exposed for any of the bacterial actions. Okay, so this is how the abscission is going to be formed. So before abscission zone is formed, which is going to form a protective layer against environment and bacterial infection. Right? Now what are the functions? One of the important functions is senescence. Again, like senescence we have already discussed earlier, it is a programmed death. Here also abscisic layer is going to be formed. So what happens? Ethylene forms abscisic layer in case of leaf, but in other parts it is the abscisic acid that forms the abscisic layer. So whenever the senescence happens, what happens? There is a degradation of the chlorophyll, and that is actually followed by the closure of the vascular connections. Right? So this is one of the most important function of ABA. So two functions, one the formation of abscisic layer and second is the senescence. Okay. There are a few more functions like promotes seed dormancy, stress hormone. Why we call them a stress hormone? Because whenever there is a environmental stress in the form of high temperature or less water, then it leads to closure of the stomats. Okay, so what actually do? It demobilize the nutrients. So once the nutrient is going to be demobilized, naturally the stomats are going to be closed. Okay. One more thing, what happens that whenever we apply gibberellin on green orange, okay, they turn yellow. Why? Because the application of ABA results into synthesis of carotenoids, and we know very well that the color of the carotenoids is yellow. Okay, so these are some of the functions of the abscisic acid. Right? So that's all as far as the inhibitors of ethylene and abscisic acid is concerned. Right? Thank you.